Welcome to this lecture on the fundamentals of genetics. Today, we are going to explore a fascinating topic known as multiple alleles. If you have ever wondered why there is such an incredible variety of traits in the natural world, from the different blood types in humans to the beautiful range of coat colors in animals, this is the secret behind it. We will build our understanding from the ground up. So even if you are completely new to genetics, you will soon see how simple and elegant these biological rules truly are. Let us dive in. To start, let us imagine you are buying a brand new car. The dealership offers this specific model in several different colors, like red, blue, green, silver, or black. Even though the factory produces 5 different paint colors for this car model, the single car you actually drive home will only have one color. Nature works in a very similar way. In basic genetics, we often learn that a gene only comes in 2 versions, like a simple light switch that is either on or off. But the real world is much more like that car dealership, offering a wide variety of options for a single trait. Building on our car analogy, let us look at how this applies to living organisms. While introductory biology might teach us that a gene only has 2 versions, perhaps a dominant and a recessive form, the reality of nature is far richer. For many traits, there are not just 2 options, but a whole menu of genetic variations available. This beautiful variety of options for a single trait is exactly what we call multiple alleles. It is the fundamental secret behind much of the biological diversity we see in the world around us, allowing species to adapt and thrive in countless different ways. Now that we have an intuitive grasp, let us define this formally. In genetic terms, multiple alleles refer to a situation where a single gene has more than 2 alternative forms existing within a population. To understand this deeply, we must remember that a gene is a specific sequence of DNA located at a precise position, known as a locus on a chromosome. In a standard diploid organism, which includes humans and most animals, an individual can only carry a maximum of 2 alleles for any given gene. You receive exactly 1 allele from your mother and exactly 1 from your father. This brings us to a crucial distinction in genetics. Because you only have 2 parents as an individual, you are strictly limited to just 2 genetic slots for any gene. However, when we zoom out and look at the entire population of that species, we might find 3, 4, or even dozens of different alleles for that exact same gene locus. Therefore, the concept of multiple alleles applies strictly to the population level, not the individual level. The individual is limited to 2 alleles, but the population as a whole holds the entire diverse pool of genetic options. Understanding this population-level diversity leads us to ask, why does this matter so much? First and foremost, multiple alleles are the primary driver of genetic variation within a species, which is absolutely essential for evolution and survival. If every gene only had 2 versions, populations would be highly vulnerable to diseases and environmental changes. In our daily lives, the most famous and critical example is the human ABO blood group system, which dictates safe blood transfusions. Another classic example is coat color in rabbits, allowing them to camouflage in environments from snowy mountains to dark forests. Without this, our rich biological tapestry would be a simple binary world. Now that we understand the biological importance, let us look at how we measure this diversity. When a population has multiple alleles for a single gene, geneticists need to know how many different genetic combinations, or genotypes, can possibly exist. We calculate this using a specific mathematical formula, which saves us from manually writing out every single combination. In this formula, the variable G represents the total number of different possible genotypes in a population. The variable N represents the total number of different alleles that exist for that gene in the population pool. As the number of alleles, N, increases, the number of possible genotypes, G, increases exponentially, creating massive genetic diversity. Let us apply this formula to a real-world scenario, the human ABO blood group system. We are given that this blood group is controlled by a single gene with 3 distinct alleles in the human population, typically written as IA, IB, and lowercase I. We need to find out how many different genotypes are possible. Since there are 3 alleles, we set our variable N equal to 3. We substitute 3 into our formula. Following the order of operations, we first add 3 and 1 inside the parentheses to get 4. Multiplying 3 by 4 gives us a numerator of 12. Finally, dividing 12 by 2 gives us our final answer of 6. This means there are exactly 6 possible genetic combinations for human blood types. Having successfully used the formula, let us take a moment to understand the logic behind its structure. Let us look at the numerator first, which is n times n plus 1. The formula requires us to multiply the total number of alleles, n, by the next consecutive integer, n plus 1. Mathematically, this step calculates the total number of pairings if we incorrectly assumed that the order of inheritance mattered. It gives us the raw, unadjusted total of combinations. You can think of this as finding the total grid size of all possible genetic pairings before we start removing the duplicates. This brings us to the denominator of our formula. Why do we divide the entire equation by 2? In genetics, inheriting an allele like IA from your mother and IB from your father results in the exact same genetic makeup as inheriting IB from your mother and IA from your father. The order simply does not matter. Because our numerator counted both of these mirror image pairings as separate events, we must divide the total by 2 to eliminate the duplicates. This crucial division step removes all redundant pairings, ensuring that each unique genotype is only counted once, making the formula biologically accurate for diploid organisms. Now that we have proven the math, let us visualize exactly what those 6 genotypes look like. On screen, you see a diagram illustrating the ABO blood group system. At the top, we have the chromosome with the specific gene locus highlighted. From this single locus, the population has 3 available alleles: IA, IB, and lowercase I. Below, you can see how these 3 alleles pair up to create the 6 unique genotypes we just calculated. Notice how the genotypes IAIA and IAI both result in blood type A, while the combination of IA and IB creates blood type AB. This perfectly illustrates how 3 alleles generate 6 genetic combinations, which then produce the 4 physical blood types we observe in humans. Let us solidify our understanding with a slightly more complex example found in nature. The coat color of rabbits is determined by a single gene that has 4 different alleles in the wild population: C for full color, CCH for chinchilla, CH for Himalayan, and lowercase c for albino. We are asked to find how many different genotypes can exist. Since there are 4 alleles, we set our variable n equal to 4. Substituting this into our formula, we first add 4 and 1 inside the parentheses to get 5. Multiplying the 4 alleles by 5 gives us a raw total of 20 pairings.
Finally, we divide by 2 to remove the duplicate mirror image pairings. Our final answer is 10. This shows that adding just one more allele significantly increases the genetic diversity to 10 possible genotypes.
We have reached the end of our journey into multiple alleles. Let us review the most important ideas. First, always remember the distinction between the individual and the population. While an individual organism is strictly limited to carrying just 2 alleles for any given gene, the population as a whole can possess a vast library of them. Second, we learned how to mathematically prove this diversity using our genotype formula, showing how a larger pool of alleles generates an exponentially larger variety of genetic combinations. Finally, this concept bridges the gap between simple genetics and the complex reality of the natural world. Multiple alleles are nature's brilliant way of ensuring populations remain diverse, adaptable, and resilient in an ever-changing environment. Great job following along.